Hello, I'm John Gribben from Bart's Cancer Institute in London. Welcome to very hot and steamy Vienna, where we're here for the European Hematology Association meeting. I'm joined by a very esteemed international panel of experts to review the highlights of the meeting. So if we'd introduce ourselves, Umberto.
I'm Umberto Vittolo from Hematology, University Hospital Città della Salute e della Scienza, Torino, Italy.
My name is Maria Victoria Mateos and I work as hematologist at the University Hospital of Salamanca in Spain.
I'm Paolo Ghia from Milano, Italy, and working at Università San Raffaele in Milano.
Okay, so what we have is a panel of experts in lymphoma, myeloma, CLL, covering the spectrum of the hematologic malignancies that we've been seeing in the meeting. So tell me, Umberto, what have you seen so far at the meeting that struck you as exciting this year?
Well, there's many, of course, topics for lymphoma in this meeting. Also, more topics will be next week because there is the International Malignant Lymphoma Conference in Lugano. But anyhow, there's a couple of interesting presentations in mantle cell lymphoma yesterday that cover the use of novel drugs. In this case, there was a first presentation made by our group in which we presented the result in young patients with high-dose chemotherapy autologous stem cell transplantation followed by maintenance with lenalidomide compared versus observation. The results so far are preliminary, so we had only the results for the first part of the study, but it's interesting that to note that the complete remission was quite high after high-dose RC, about 77%, along with the MRD negativity, but with additional autologous stem cell transplantation, it doesn't seem to increase so much. So I think because only a few points more after high-dose chemotherapy. So I think this open a window regarding if we can spare high-dose chemotherapy, maybe autologous stem cell transplant in this patient, maybe with addition of novel drug like lenalidomide in the maintenance or ibrutinib in combination with chemotherapy. There are some ongoing studies, ongoing trials that aimed at this point, just adding novel drugs to chemotherapy backbone plus, of course, anti-CD20 antibodies in order to spur a more aggressive treatment that usually is required for the treatment of mantle cell lymphoma.
Are these minimal residual disease endpoint studies? Have they built assessment of MRD into the design of the study?
Yes, MRD was a secondary point of the study, but it was assessed after each step of the treatment, after the first courses of R-CHOP, standard R-CHOP, then after high-dose chemotherapy with RC before autologous stem cell transplant, and during the maintenance. The data so far we have is in quantity PCR was 70% of MRD negativity before autologous stem cell transplant, and after autologous stem cell transplant 72%. So it doesn't seem
to add so much for the toxicity.
And we'll see in the future during the maintenance period.
So watch this space for the follow-up of this study is basically where we are at this point.
The maintenance is planned for 2 years, and I hope that we have the data for the maintenance next year. Okay. Next year, of course.
Okay. And in terms of myeloma, lots of new drugs emerging in this field. Of course, for those of us who aren't myeloma experts, look at it and think, how on earth do you start deciding which of these so many new agents appearing are going to be the ones going forward? So what's your view of where the field's going based upon what we've heard here?
So absolutely, novel agents are coming for all hematological diseases and also for myeloma. So from this European Hematology Association meeting, I would like to highlight novel agents, second-generation proteasome inhibitors, and carfilzomib has demonstrated in a large randomized trial to be superior to bortezomib plus dexamethasone with a significant benefit in terms of overall response rate, complete remission rate, and also duplication of the median progression-free survival.
with less toxicity, or is the toxicity similar?
Toxicity is it's different because carfilzomib has a specific toxicity profile. Carfilzomib does not induce peripheral neuropathy as occurred with bortezomib.
But
however,
it has been a big problem for patients to tolerate. So do you find it better tolerated?
Yes, I think that definitely the tolerability is better, although we have to be careful with some side effects such as renal impairment or cardiac toxicity, but in a specific subgroup of patients.
Sure.
I would like also to highlight novel agents such as HDAC inhibitors that they have been evaluated also in randomized trials, and panovinostat plus bortezomib and dexamethasone showed to be superior to bortezomib plus dexamethasone. But what I would like to highlight of this EHA meeting is that sub-analyses have been conducted in patients who had been previously exposed to bortezomib and IMiDs. And in this specific subgroup of patients, heavily pretreated patients, the difference, the benefit in median progression-free survival has been much more evident, with a median progression-free survival benefit of approximately 7 months for panovinostat, bortezomib and dexamethasone, confirming the role of HDAC inhibitors in the management of myeloma patients. And finally, I would like to highlight the immunotherapy strategies through monoclonal antibodies. Today, we have had the opportunity to hear from Sagarlonial from the US the results of daratumumab single agent in heavily pretreated myeloma patients, exhausted to all conventional agents, bortezomib, thalidomide, lenalidomide, carfilzomib, and pomalidomide, and this patient responded to daratumumab single agent, emerging as a possible backbone for all regimes in the treatment of myeloma patients.
Sure. And accessible to younger and older patients.
Yes, results are very preliminary, only in 100 patients, but it seems not having differences according to the age.
Sure. And of course in CLL, lots more on the novel agents and follow-up. So what struck you in CLL that you've heard from this meeting today?
Yeah, now the novel agents are a reality in CLL since a couple of years at least. So I would say that this meeting in particular has been like a consolidation phase. So we saw the long-term results of the use of ibrutinib or idelalisib plus rituximab. And indeed, they confirm durable responses. They confirm also the toxicity profile that we are now aware of, and in particular the diarrhea that is quite frequent, around 40% in patients treated with idelalisib in the long run. New molecules have been presented also of the same family, I would say. So another PI3 kinase delta inhibitor, TG1202, that apparently has less toxicity.
The toxicity does seem less, less GI toxicity, less hepatotoxicity.
It is true that the follow-up is only 6
short,
months, but still only 2% apparently, so that would be good news. And then we saw also the results of the PI3 kinase gamma delta, so the dual inhibitors also on CLL and other malignancies, and the results are also quite in line with what we learned about idelalisib. And of course, what we are waiting for are the long-term results of the BCL2 inhibitors, the ABT199. Here, the Australian colleagues show the results after 30 months of follow-up of even patients who's in complete remission who stopped the drug because of any reason, and they showed durable responses even outside of therapy. And they confirmed over 40% of complete remissions with a very high number, around almost 50% of patients with MRD, of the patients responding. So these are promising results, and so the future appears to be quite bright.
Sure. I mean, I think the ABT studies showing the MRD negativity look really intriguing, because one of the things I was going to come back to talk to all of you about are all of these novel agents greatly increase the potential cost. And certainly in Europe, we have to be thinking about the cost to our patients. And I guess I was asking about MRD in the mantle cell setting. You certainly look at that a lot in myeloma and now in CLL. But therapy that potentially has a beginning and end, is probably where we're looking to go forward, I think, in all of these diseases, rather than continuous therapy. Do you agree?
No, if I can answer you, I think you're totally right, because there's a couple of issues. The first one, as you remember, the issue of the cost, that is because all these drugs probably are high costly, so it's difficult to think that we can give to a patient for a life. The second point may be that we don't know exactly yet the long-term toxicity of the drug, because all these agents are active on B cells, B cell receptors, pathway, and so on. So we don't know exactly after some years of exposure how will be the immune functions of our patient. Of course, the first goal is to cure the patients. That's we need to cure probably in the relapse setting. But I believe that we need to know more on these drugs, and also to try to design smarter studies in order to fix an end to this treatment. Maybe like in CML, as you know, in CML, after some years of complete molecular remission, we are thinking now to stop the TYK inhibitors, and maybe also in lymphoma and in CLL. I don't know if myeloma, but we should think something similar to CML.
Of course, in myeloma now, we're seeing, because you have so many agents available, people going through 7, 8, 9 lines of therapy. It's becoming increasingly common to see patients surviving a very long period of time. But of course, we also see that patients are sometimes surviving a very long period of time with myeloma with still having increasing morbidity from the skeletal complications of the disease. Are there ways in which we think that some of these new agents are going to impact the long-term quality of life of your patients?
Yeah, I think that it is necessary to do quality of life studies in all trials that we are going to conduct, because you are right that the survival of our myeloma patient is clearly increasing. In fact, with the daratumumab, 60% of the patients remain alive after 1 year, and this is surprising because in the past these patients were not alive more than 9 or 10 months. So I think that our efforts have to be toward that direction, not only to survive, to be alive more time, but to try to improve the quality of life, of course, because the bone disease is one important issue in myeloma patients, and so we have to try to control and to improve this symptomatology.
Sure. So Paolo, of course, in CLL, the approved agents that we have are continuous therapy at very high potential cost. Do you also believe that the future has to be to find a way to get these patients into a status where we can potentially stop the drug? Or
yeah, definitely yes, and indeed the next generation of clinical trials will be combination of the drugs. So we understood that with these new products we cannot achieve a complete remission, or at least the vast majority of patients. So the future is very likely to combine with something else, and ABT-199 is probably one of the best candidates. Studies are starting or are designed to combine with either ibrutinib or idelalisib, and or duvelisib even. So this is the way in order to reach more profound responses, MRD negative responses, and especially to stop after a definite time the treatment.
The idea, of course, would be that even though the cost is 3 times as much because you combine 3 agents, it's at
correct.
least for a defined period of time, and then you can stop.
Yes,
right?
then of course, I mean, you spend a lot, but if that is for 6 months, as we were used to do with the chemotherapy, then probably it's more affordable than just having no limits in the future.
Okay, so what you are hearing from Vienna is continued excitement that in the hematologic malignancies there's a huge number of novel agents which are being used alone and in combination, looking to improve the duration of responses, the quality of life, applicable to younger and older patients in a way that chemotherapy wasn't in the past. So these remain very exciting times in terms of the management of hematologic malignancies.